Hey guys, Mr. P. In this video, we're going to talk about the interphase portion of the cell cycle. We're going to focus on the IB understandings D215 and D2114. D215 states that DNA replication is a prerequisite for both mitosis and meiosis, and therefore happens in the interphase. Students should understand that after replication, each chromosome consists of two elongated DNA molecules called chromatids, which are held together by a common centromere. D2114 states that cells grow during interphase and that students should appreciate that interphase is a metabolically active period and that growth involves biosynthesis of cell components including proteins and DNA. Number of mitochondria and chloroplasts are increased by growth and division of these organelles. So, in this particular video, which is going to be a very short and targeted video, specifically about these two understandings as it relates to the metabolic processes that happen during interphase. So the first thing we need to do is kind of do a recap of the cell cycle. Now, in the last couple of videos, we've talked about what happens in the cell cycle, what the cell cycle is. If you haven't seen those, or if you need a refresher, go back and watch. But for now, we know that interphase, which is this blue section, consists of G1, S, and G2. G1 stands for gap1, S stands for synthesis, and G2 stands for gap2. This particular video is going to focus on the interphase portion, so what happens in the cell cycle from the beginning of G1 to the end of G2. So if we focus on the S phase, which happens to be right in the middle, and one of the IB understandings for this particular video, cell division cannot happen until the DNA is replicated during S phase. So we know that S stands for synthesis, and this is a portion of the cell cycle in which DNA replication takes place. The cell is not able to fully travel the entire cell cycle and therefore divide itself unless the DNA is replicated first. That DNA replication takes place during this S phase. And so how does the DNA replicate itself? Well, at the beginning of S phase, these chromosomes, which consist of long strands of nucleotides, are going to be non-duplicated. So we will say these are non-duplicated or non-replicated chromosomes. These chromosomes are said to be homologous chromosomes, meaning you have one chromosome from dad and one chromosome from mom. They are not identical, however, they contain the same genes. And so if this particular chromosome from mom has a certain number of genes, the father's chromosome, which is homologous to the mother's chromosome, will have the same number and location of genes, but the alleles that are contained within these genes may differ. And we'll get more into this when we talk about genetics. But for instance, the mom might give you a dominant A and the dad might give you a recessive A. These chromosomes are going to duplicate themselves. Notice they go from a single strand to a double strand held together by a centromere. These homologous chromosomes, again, one of the chromosomes is from dad, one of the chromosomes is from mom. They are homologous chromosomes. They have the same number of genes, same gene locations, but they have different alleles. They need to not only duplicate themselves, but they need to help prepare the nucleus, and they need to become prepared themselves in order for the cell to divide appropriately. So the replicated DNA is arranged in a chromosome as two sister chromatids. The sister chromatids are exact copies of themselves. Notice the green chromosome duplicated itself and became a second green chromatid. Those two chromatids are said to be sister chromatids, and they are held together by this central centromere, which is holding the two sister chromatids together. The paternal sister chromatids are also held together by a centromere, and that allows for the sisters to stay conjoined. Two sister chromatids are held together by a centromere. And why do we need to supercoil or condense the chromatin into these particular active forms of DNA, which are called chromosomes? It's because during interphase, the DNA is inactive, but duplicates itself and eventually becomes the X-shaped active form of DNA called chromosomes during M phase, which will be a lot easier for the cell to move around and ultimately divide. Each of the new daughter nuclei, before the cell divides via cytokinesis, will package the recently divided chromosomes into a new nucleus and will split the cytoplasm, forming two daughter cells from one. During division, the two sister chromatids are pulled apart. At that moment, they each become an individual chromosome, one for each daughter cell, like they were before the S phase. So they are duplicated during S phase. They will then go through the rest of the cell cycle. They will divide themselves. They will be packaged into two new daughter nuclei, where they become a unduplicated chromosome, which will enter the next cell cycle and will eventually be duplicated within their own S phase. Cell growth during interphase, remember, contains or is contained within the G1 or the gap1 phase, which again stands for gap but has a lot of cell growth in it. At the beginning of the G1, the cell is the smallest it will ever be, and so that cell starts out really, really small. Through the G1 phase, it will continually grow. It accumulates materials such as proteins and nucleotides, and will end the G1 phase as a much larger cell than it began. Remember, G0 is a kind of standalone separate process which occurs when the cell needs to enter a state of stasis. However, if G0 is bypassed and the cell is allowed to go through its normal G1, it will start small and end large. As it grows, it accumulates these proteins and nucleotides. What are the proteins and nucleotides going to be used for? Proteins are going to ultimately be used to manufacture structural components and functional components the cell needs in order to function correctly, and then nucleotides help to produce the building blocks of the nucleic acids, which will be the DNA that has to be synthesized in the S phase, which again is DNA replication. If we focus now on the G2 phase, additional growth is going to happen. Notice the cell starts G2 much bigger than it started G1. That's because a lot of the cell growth happened during G1, and so by the time the cell grows during G1, it duplicates its DNA and enters G2. It is a much larger cell. It still will, however, grow a little bit more. Once the chromosomes have been replicated, the cell enters its second growth phase called G2. Organelles such as mitochondria and chloroplasts can increase in number. DNA begins to condense from chromatin to chromosomes, and the microtubules can begin to form. All of those things have to happen in preparation for the cell division phase, which is mitosis, and the cell again gets physically larger on its way to M phase. When it enters M phase, which is this portion of the cell cycle right here, the cell will be as large as it will ever be. Through M phase, which is a cell division, this large cell will divide and will produce the two small cells, which again entered the next round of the cell cycle, which is G1. That's it for this particular video. Again, it was a very targeted video, specifically about the stages and functions of the G1, S, and G2, which make up interphase collectively. If you learned something, give it a thumbs up, subscribe to the channel. We'll see ya.